Hello everyone. So in the last week, you have studied about the different applications of optical microscopy and electron microscopy. So I'll be giving a brief about the applications of the optical microscope and AFM, and I'll discuss the basics of SEM, TEM, and their applications. So let's start with the applications of optical microscope. So, just to give a quick overview, um, in the optical microscope, we have a objective lens. Uh, sorry. So, in the optical microscope, we have an objective lens and an eyepiece. So, these two lenses actually magnifies the image with the help of these two lenses, and we get a bigger uh, image. So basically the magnification happens with the help of objective lens, and with the eyepiece lens we are able to see the image. So then other types of microscope is the confocal microscope and phase contrast microscope. So in the confocal microscope, we use a dichroic mirror and a laser as a source of light is used. So in we have different excitation filters for the laser. So depending upon our choice of excitation, we apply the filter over here, and then with the help of dichroic mirror, what it does that it blocks the reflected light to the camera. So here, if you are exciting with suppose green laser, or uh, green is 500 nanometers around wavelength. So the reflected green will not go from this dichroic mirror, but only the excited uh, uh, laser. Suppose if the material is absorbing the green color and emitting the red color, then only the red color will pass from the mirror. So that is how the confocal microscope works. Then we have the phase contrast microscope. So in this we use a this annulus aperture and a phase plate. So with the help of these two, we get the contrast in the interference. So if there is a constructive interference, we will get a bright image, and if there is a destructive interference, then we will see dark image. So with the help of this, we are able to get the phase contrast in the microscope. So this was about it. Then coming to the application. So suppose if we want to see the morphology of a fiber. So if we see in the optical microscope, we will see this long fiber like structure. But if we want to see the magnification, high magnification image of this, so then again we have to go to the with high objective lens. So suppose this is at 10x magnification, this is at around maybe 100x, since it is not mentioned, so I'm just guessing. So it is like 10 times more of the magnification. So as you can see from the scale bar also, this is 10 micrometers, and if when we zoom in, it becomes 1 mm. So likewise, this is how the uh, microscope we can use for the various applications. So then there are the fiber cross sections also we can see. So we can just cut the fiber cross section, and depending upon the morphology, we are able to see different morphologies of the fiber. So the first one is the round shaped polyamide. Then the second one is the irregular shaped pan, that is polyacrylonitrile. Then we have this trilobular shaped Orion, and then this is the dog bone shaped Orion fiber. So these are the fiber sections. So cross sections also we can observe in the optical microscope. Then for composites also we can observe in this optical microscope. So first is the mica flakes. which are this plate kind of or sheet kind of structure morphology, and the shape is very irregular, so that we can observe. And the second one is the talc powder, talc particle, having very fine and fine texture, and its shape and size is also very like wide range of shape and size it is having. Then the third one is the clay particle, which are very fine particles. So there is no characteristic shape boundary for this. We can observe in the microscope. So for these kind of structure, then we have to go to higher magnification. So then we will use the electron microscope. Then for different fibers and films, we can observe in this. So this is a polyethylene fiber cross section. It reveals a very fine and spherical texture. And here we have a spherical texture in again in the polyester film cross section. So these two are the cross sections of the fiber. And then we have this. So orientation of the electric field, this is also can be observed. So this material actually gets stimulated with the help of electric field. So the orientation of these fibers or this material, this gets bent when we apply the electric field, so that we can observe uh, with the help of the optical microscope. Then texture of the extruded film. So suppose we are extruding a sample and we want to check how the morphology is taking place when upon extrusion. So that we can observe with the help of optical microscope. So this is on a TEM grid. Uh, we are observing the very cross, very thin longitudinal section of this polymer. And so these hexagons are because of the TEM grid. And this fiber is at is very thin slice of this fiber is kept on the grid. And in the bright region we can observe that the fiber is present. And so the skin of the fiber appears very bright yellow, and the core appears bluish. And that is less oriented, but this is highly oriented, as you can observe here. Then the structure of molded nylons, that is also can be observed with this polarized light microscope. So this is the top of the a nylon. And so this is showing a very round this round spheroid in the transition zone we can observe. So this radial kind of texture, these are all visible in the light microscope. So this is a cross section of a polyacetal polymer, and we can see the spherical texture here also. And the so this is the bifringent skin uh, of the uh, of the material. And the and an unoriented core. So this is the core, which is not at all oriented, but and this is the skin, and this is the outer node, like that. And this is showing between the skin and the core. In the transition zone we can see here. This is the image of the from the transition zone. And the third is the spherulites that are polygonal in shape, which is which can be seen in this figure. Then for as contrast, optical microscope can give us the impact modified nylon. So it is showing a very fine dispersion of modifier in the matrix. So as you can see here, this is the modified nylon, and you can see the fine dispersion of the modifier. Now coming to the applications of the atomic force microscopy. So uh, just before the application, I'll be giving a very quick recap. So AFM, we have this cantilever with the tip, and it is attached, and the deflection is observed. So the deflection of the cantilever beam is observed with the help of a laser. So on top of the cantilever, we have a very smooth surface, mirror-like surface, and the laser falls over it and goes to the mirror. And from here, here we have a position sensitive detector, that is what detects the position of the cantilever. So the up and down movement of the cantilever with the tip is observed, and so this position center of sensitive sensor of this observe the deflection of the beam, this cantilever beam, and with the help of the control unit, it comes back to its original position. So that get processed in the system, and then we are able to observe the topography of the material. So this top layer we can observe. So this can be of three types: contact mode, in which the tip is in contact with the sample. Then the non-contact mode, in this the tip is not in contact, but it is kept at a uh, constant height from the sample. So if the height varies, then the cantilever moves. And the third one is the tapping mode. So here it moves in a fashion like it touches the surface and then comes back, and then again then touches the sample and comes back. Like this it will go. So this is called a tapping mode. And as I told, this is the position sensitive detector. It is a quadrant photodiode. So in this four quadrant, suppose the beam is getting deflected, then we will observe that it is moving upward or downward or left or right. So with the help of that deflection, we are able to detect the morphology. So in the AFM, we are able to observe the topography, as I told. So suppose this is a fiber. So the fiber topography can be observed, and when we go to a higher magnification, we are able to see the very distinct phases. So suppose that this fiber is having a cellulose and lignin. So here we can see the structures of cellulose and lignin very differentially. So those kind of structures can be observed in the AFM. So in the AFM, we can also chemically modify the cantilever tip also. So that the attraction or repulsion of the AFM tip can be enhanced, and with the help of that, we can observe the changes happening in our biological system. So this is generally done for the biological samples. So suppose AFM tip is functionalized with a certain kind of receptor, which attracts the sample. So with the help of that, we can observe the changes in the morphology. Then this is the AFM of a spider silk fiber. So as you can see, the morphology of this fiber is very well seen with the help of the AFM, and the height is only 200 nanometers. So this kind of small morphologies we can observe in this AFM. And this is a crystal structure of a polymer. So you can see the scale bar here, 100 nanometers. So up to this dimension, we can observe the orientation of the grains and all in our polymer. So this shows a very lamellar kind of a structure for the polyethylene film, and this so with the help of this, we can observe how much will be the depth and all of the film, that can also be observed from the AFM. Then for the surface morphology after processing and before processing, we can compare. So since AFM is basically used for the surface based characterization, so as you can see here, we have this dielectric thin film. This is benzoic butadiene (BCB). This dielectric material is modified later with the help of oxygen plasma treatment was done on top of that to remove the residues. So this both before and after images can be compared uh, with the help of the AFM characterization. Then we can also observe the copolymer morphologies. So suppose we are adding two polymers or three polymers. So those So suppose we are adding three three block polymers, tri-block copolymer, and on a surface of a silicon substrate, and then we are annealing it, and we'll observe how will be the phases change, or the height of the film is changing. So that also we can observe from the AFM. This shows the height of the uh, block polymer, and this shows the phase. What are the phases present? Then the addition performance can also be checked in the in this kind of with this kind of characterization. So in the intermittent contact, that is the tapping mode, this image was taken, and the left one is the spin coating at room temperature, and after baking on the hot plate, they are at 100 degrees centigrade. They again check the morphology. So this shows the addition performance of our sample. Then strain induced structural changes. So here, so when we apply the strain on a material, the structure of the crystal structure will change for the sample. So those changes can also be observed. So this is the cross section of this PETG film, and so it is having different layer thickness at different so these three samples is having three different layers thickness. First one is having 60 nanometer thickness, then center one is having 30 nanometers, and the right one is having 8 nanometers. So those kind of very little thickness can also be observed in the AFM. Then the membrane morphology can also be checked with the help of AFM. The roughness, surface roughness, and other parameters can be checked to get the fouling properties or of the membrane, uh, and other the surface morphology that can be observed. Then, if there are micropores present, so this is a microporous membrane. Here you can see the it is showing like the small pores are visible over here. This is on the height, so you can see the heights of these pores which are present in the membrane. So that can be analyzed. Then this is a glass fiber image. So the first image shows the uh, image of a glass fiber having heat cleaned surface and acid treated, and this shows a very little surface detail. And then they analyze the fiber, glass fiber, so it is showing the details with the height also. And then this is the after treatment with acetonitrile solvent, so that details of the particle size and all we are able to see in these pictures. Then the thermoplastic vulcanization, this kind of phases after vulcanization, this uh, in the thermoplastic, the uh, these phases will be very much distinct when we look in the AFM. So this is the height morphology, this is the phase contrast. So this is very well visible in this images. Then we have the banded spherulite kind of polymer. So this is a very high density copolymer. So when we observe the 3D stacking of this image, we can get the height of this our structure, and then the morphology is also very well seen. Then the intermittent contact mode, again tapping mode. They have taken this kind of images of a LDP pellet. So this is low density polymer, and so this is taken before and after the permanganate etching. And this is before and this is after the etching. And the C image shows the spherulites size. So it is around 20 micrometers on the side. So this is also very clearly visible. And there are debris on the original surface. So those kind of details uh, can be observed. And the in the etching, after the etching, the the debris were removed. So we can see the clear image of this of our LDP palette. Then I'll be giving a brief on the electron microscope. So electron microscope is a technique which uses a beam of accelerated electrons as a source of illumination. So up till here we were using the optical microscope, in which we were using light as a source. So the normal visible light we were observing. Then in the AFM we were using a probe, and in the electron microscopy we are using a accelerated beam of electrons as a source of illumination. So these are again classified into two types. This SEM, that is scanning electron microscope, and TEM, that is transmission electron microscope. So starting with the SEM, so suppose I'll be giving a brief overview on the structure and morphology. So suppose we want to check the electronic levels or subatomic level. So with the help of TEM we can observe that. Then atomic level, molecular level, or chemical composition, this also we can observe with the help of TEM. Then crystal arrangement of atoms or ions with respect to one another. This kind of arrangement of atoms can also be seen in the TEM. So TEM is a very highly uh, very high resolution technique. Then we can see the microstructure. It can be also seen from the optical microscope, or we can see from the SEM image also, the microstructure of a material. And macrostructures we can see from the naked eye also. So electronic and atomic structures are around 1 angstrom levels, and then the crystal structures are around 10 angstroms. In the X-ray diffraction, we can see those structures, or even in the TEM, we can observe them. Then microstructure, that is from 10 to 10 to the power 6 angstroms. This we can see from the electron microscope, and also from the optical microscopes sometimes. Then the macrostructures can be seen from magnifying glass, or with naked eye also. So these polymer structures are having different regions. When we are talking about a polymer, we are having lot of different domains. So suppose we have a interlaminar or amorphous region, then we have a crystallized domain, that is crystalline. Then we have spheroid boundaries. Then we have filler particles that has very low interfacial adhesion. Then we have some micro pores or voids also, or cracks also sometimes. Then we have phase separation, so different phases can also be present in our material. So here, as you can see here, this is a chain end, this is a weak entanglement, then this is a overstressed bond, this is a weak cross-linking, this is a interlaminar defect. So these kind of observations can also be done with the help of electron microscopy. So as I told that different regions are having different resolutions. So the grains and powder particles, these we can see with our naked eye also, or also in the microscope, optical microscope. Then we have the powder particles, then and again spherulites. This kind of this we can see from the optical microscope. Then these are two phase structures and lamellar kind of structure. This you can see from the scanning electron microscope. And then the crystalline structure and molecular structures. For this, we have to go to the transmission electron microscope. So different techniques are having different resolutions. So as I already told you, the light scattering one is having very low resolution, and optical also. Then as we move towards the electron microscopic electron based, we are getting higher resolution. So for the TEM, it is around zero point 0.2 nanometers. And then as we move towards the X-ray based techniques, we are getting also very high resolution. So this is a complete overview of the all of the um, microscopy techniques which are present for the characterization of polymers. So the structure and property correlation we can observe with the help of uh, microscopy or electron microscopy. So suppose if in a polymer we are having a molecular structure, and if we do some processing in it, or some loading condition, if we want to check the mechanical property of the system, so those kind of this actually affect the property of a material. So suppose if I am adding a filler in the polymer in the main polymer structure, so in that case the voids will be lesser, so its ductility will increase. So the property will change and its structure will also change. So that is why it is very important to analyze the structure of a material. And also, so if I put some load on the polymer, and it got deformed or fractured, so how it is deforming and what kind of fracture it is producing, those kind of analysis gives me the idea about the property of the material. So those kind of things are important when we are talking about the polymer. So the morphology is very important. It gives the correlation with the property of a material. So that's why we have to check the morphology of the material. Then, so there are basically four principles of the microscopy. So first one is the transmitted beam, which in the light microscope or optical microscope we have used the transmission mode, and the TEM also uses the same transmission. Electron microscope also uses the same principle of transmitted beam. Then we have a reflecting beam. So when we are putting the electrons on the surface, it gets reflected back. So sometimes in the light microscope we uses the reflection mode also. So this this radiation will be observed, and in the electron mirror microscope this mode is used. Then we have the scanning beam. So this beam actually scans with pixel by pixel and gives us the image. So this kind of principle was is used in the scanning electron microscope and the confocal laser scanning microscope. And then we have a scanning tip. So as I told already, in the AFM we have a probe, and it goes in the XY direction and Z direction. So this principle is used in the scanning tunneling microscope and AFM, that is atomic force microscope. Then uh, So this is a basic difference between all these kind of microscopy. So the SEM gives us the surface morphology again, and if you want to check the bulk, then we have to cut the surface, and the cross section we will get that can be easily observed in the SEM. Then STM will solely give the idea about the surface only. Then transmission again can give us the morphology of the surface or the bulk if we cut it in a very thin cross section. That that material can be used for the TEM analysis. And then we have this X-ray based. So this is again from the bulk. If we cut a sample, that we can analyze using the X-rays. So this is a normal television monitor, and it uses the same electron gun which we use to generate the electron beam for the microscopy of our material, the electron microscopy technique. So we have electron gun, and here we have magnets to actually um, actually make the beam focused, and this is the electron beam. So it the magnets actually helps to point the beam in at a certain position, and it occurs in vacuum because we do not want any interaction of the electron with air. If it interacts with the air, then it will get deflected in all the directions, and we are not able to get a very highly pointed or focused beam. So that's why we have to use this in vacuum. And then we have a screen which is a phosphor coated screen. So this displays our image. So now let's look at the scanning electron microscopy and its setup. So here we have an electron gun, and with the help of these lenses, magnetic lenses, here we have a anode, and from the electron gun, the electrons are coming towards the anode, and this is the beam. So with the help of a magnetic lens, we are able to align the electrons in a parallel line, and then we have a scanning coil, and then backscattered electron detector, and then finally, so with the help of the magnetic lens, these are coming to the as a pointed beam, focused beam on the surface. Then the scanning coil goes to the connector or the TV scanner. So this is the actually the screen of our system. Then the backscatter detector detects the backscattered electron. So and the secondary electron detector detects the secondary electron. So what are these electrons, backscattered and secondary? So I have already mentioned you in the previous class, but I'll again quickly give a review, overview. So when we incident a beam on a sample, so some of the rays will get or the electron will get diffracted, scattered, or diffracted after getting collided with the sample. So if the angle is very less, those are called backscattered electrons. So these give the information about the atomic number and topographic information. Then we have a cathodoluminescence, that is the electrical information. It gives the information about the electrical property of the material. Then we have a secondary electron. This will give again the topographical information of the sample. And then we have agar electron. Agar electron gives the surface sensitivity and compositional information, and the X-rays. So characteristic X-rays give the information about the elements which are present in our sample. So the composition again, and the thickness also. So it goes in the bulk. So that is why it will give the thorough thickness information of the sample. So what are how these are generated? So the secondary electron. So suppose the electron is coming and it is knocking the one of the electrons, and this knocked out electron is getting detected by the secondary electron detector. And the auger electron is what? So when the core electron is getting uh, knocked out, then the outer shell electron will fill this gap, and it will cause it will and another one more electron is getting knocked out. This is called as a auger electron. Then we have a fluorescent X-ray. So this is the characteristic X-ray which when uh, so when we are knocking out the core shell electron, one of the electron from the other shell will jump to the to this to fill this gap, and while jumping it will emit some of its energy. So that energy it is detected. So this it releases the energy in a form of X-ray, and that is detected by the help of a detector. So that information we can get from the fluorescent X-ray microscope, and then we will be able to know that if so particular element is having a particular characteristic X-ray associated with it. So with the help of that characteristic X-ray, we are able to uh, say like which element can be present in the material. Then the backscattered electron. So suppose electron is falling towards the material, but it is not uh, colliding with any of the electron. It is not knocking out, but it is simply going back. So that is the backscattered electron. Then the characteristic X-ray, as I already told you. So these kind of X-rays, if it is emitting in a different wavelength, then we have to use the fluorescent X-ray. But if it is deflecting in the same, if it is emitting in the same wavelength, then we call it as characteristic X-ray (EDS). So with the help of EDS detector, we are able to detect this signal. Then the continuum X-ray. So the continuum X-ray is in which we are continuously getting a X-ray. Actually, the X-ray emission from the sample. So the possible types of interaction is I already told you. Secondary electron. This will give us the. This is actually a low energy electron from the atom, and this will not give us very. So this low energy electron. Why it is low energy? Because it is colliding with one of the atoms, and it is coming back. So that electron is having very less energy, but it will give a very good resolution. Then we have a backscattered electron. So that is again the collided one, but it is, uh, as I told you, the backscattered electron. It is not colliding with any of the electron or atom from the atom, but it is simply getting backscattered. So this will also give the information about the topography, but it will not be very good resolution. Then we have the backscatter diffraction. So that is the X-ray diffraction. We can that will provide the information about the spacing between two atoms. Then in very rare cases we will get the Auger electrons. So these are also used for the information of the uh, quantum energy or quantum states, but it will not usually detected in the SCM. And then the characteristic X-rays, these are detected with the help of dispersive X-ray diffraction (EDS), and this can be continuum or fluorescent X-rays also. And this EDS are this kind of detector is attached to the SEM normally to detect the characteristic X-rays to get the elemental composition of the material. So the SEM will give us. So this has different detectors in it. So it has a backscatter detector which gives us the compositional contrast. Then the transmitted electron imaging it can also be done. This gives the internal ultrastructure information. Then we have the energy dispersive X-ray spectrometry (EDS) that gives us the elemental composition or mapping and line scan. Then we have the electron backscattered electron diffraction (EBSD), that is that gives us the sorry that gives us the information about the crystal structure. Then we have the secondary electron imaging, which will give surface topography, morphology, and the particle size. So normally we use SEM so for secondary electron and backscattered electron, and also the EDS for elemental mapping. And this transmitted electron imaging is rarely used, but EBSD is also widely used when we want to check the crystal structure of a material, of a metal. Normally for metals they use it. So, field emission. One more type is the field emission SEM. So that is FESEM or FEGSEM, field emission gun. So the only difference in the field emission and the normal SEM is the this electron gun. So in this FESEM, we use a field emission gun that gives us very advanced. That is a very advanced technology, and it gives a very high resolution image of the material, because in this we are having a very like we are getting lot of electrons. So in the FESM, what happens that the electron beam size is very small, so that the resolution increases because the pixel size now is very small, so the number of pixels increases, so that's why the resolution will be higher. So now coming to the electron source. So in the SEM, a thermoionic electron source such as a tungsten filament is usually used. The cold field emission electron microscopy uses a cold cathode field emitter composed of a single crystal of tungsten etched to a very fine point. Then in SEM, the escapes from the source once sufficient heat energy has been applied to exceed the energy potential barrier. In cold FeSEM, the electric field is applied to the tip of the electron source, which is essentially is pulling the electron from the emitter, and the electrons are then accelerated off on the source. So this kind of tip that electrons will be formed. So this is the extraction anode, and this is accelerating anode. And so the electrons are then accelerated off the source by two anodes. These are two anodes. And because of the microscopic size of the electron source, the beam produced by this emitter is one of the smallest available. So since we are using a microscopic size of the electron source, so that's why the beam, as I already told you, it is very very small, and that's why it will give a very high resolution of the instrument. So SCM versus FESCM. So the biggest difference is the electron generation system. That is the FEG gun, field emission gun, is present in the FESCM. And so in the FEG, a potential gradient is applied to emit the electron beam, while in SEM, a thermoionic emission is applied. So this is the sole difference. So advantages of SEM with FEG SEM is that it gives a much brighter electron source. It has a much brighter electron source, and it has a very small beam size. So beam is very very pointed, and that's why we'll get a very good in magnification also and resolution also. Then we have environmental scanning electron microscopy, ESEM. So this ESEM is used for generally for wet samples or biological samples, because in those cases we have to have a we cannot these cannot be observed in the vacuum. We have to have a very water kind of environment for those kind of samples. So that's why these ESEM helps us to monitor the wet and uncoated samples also sometimes. Uh, so this instrument was originally designed by Gerasimos Denalitos and in the at the University of South Wales, New South Wales. So this is the typical ESEM setup, and here we do not so still the vacuum is present in the SEM. So in this column there will be vacuum, but the sample where we keep the sample, that portion is uh, not does not uh, uses much of the vacuum. So there we can keep the wet and uncoated samples. So there are various applications for SEM. So it can be used to check the material, the conductivity of a material. So suppose if a material is not conductive, in those cases we have to apply a very thin metallic film of suppose carbon or gold or silver or platinum sometimes. So platinum and gold and carbon are normally used as a metal to for the coating of the sample. And if we want to observe a biological sample like insects and all, those has to be coated first when we have to put in the SEM. Otherwise it will not give us signal, because we have to have a conductive sample for the SEM. And but still, if we cannot coat some samples, then we have to go towards the low vacuum mode. In those cases we can use the uh, biological samples, but still in the low vacuum mode also we have to dehydrate the sample. So normally for the biological samples we study in the ESEM only. Then SEM is a very extremely versatile tool which is relatively easy to learn and to prepare samples for, and compared to TM, because TM is a highly it uses a transmitting light or electron. So those in those cases we have to take care of the sample preparation very much. We have to have a very thin, ultra thin sample in order to get the proper signal out of it. So in the field of material science, it is used for the detection of chemical composition, morphology, topography, and microstructure. So SEM is used to inspect semiconductors also and microchip assemblies also. Then forensic science for in the currency examination, handwriting analysis, art restoration, etc. And then biology for insect parts, animal tissue, or even bacteria and viruses can be imaged in the SEM. So now coming towards the application. So this is showing a rubber sample and a polymer sample. So this kind of structure, uh, it shows the toughening of epoxies. So as you can see here, there are voids present, and so this is a coarse texture of cell walls of tire, which is having porosity in it, and then they have done some processing, and then it got uh, toughened. So these are different kind of images for different products. Then the dispersed particle phase particles are observed in the secondary electron imaging of the izod fracture surface of the copolymer, and this is showing about 0.5 to 2 micron dispersed phase particles. These are the particles across all over the sample, and the holes are also can be observed from in the material. And this is a dot mapping. So the characteristic X-rays are generally shown as a dot map, EDX dot map, and with the help of these dots, we can see that at these places, these are the elements which are present. Suppose this dot is for carbon, so at the dots all over the place, the carbon is present. It is just for example. Then here we have a SCM. This is secondary electron imaging, then backscattered electron imaging, and the elemental mapping. So the first one is showing the secondary electron image of a multi-phase polymer. It shows a very large dispersed phase particles and sub-micrometer size particles and holes. So these are the holes. And then we have the backscattered image. It shows, so you can clearly compare the secondary electron image shows a better resolution as compared to the backscattered electron. So this is again showing the dispersed phase particles with higher atomic number, and then the matrix. Then we have the elemental mapping. So this shows the small particles contain antimony. So in this particular sample, we are having antimony also. So that we have, this is the antimony Sb. Uh, the for the so the EDEX dot mapping is shown here for the antimony, and this is for the chlorine. So Cl is shown here. So the chlorine is present at this part mostly, so that we can observe with the help of the EDEX mapping. So we can clearly see at what point the what kind of element is present, dominantly present in the material. Now coming to the TEM setup. So TEM is called as transmission electron microscope, and it uses the, as I told here, we have to uh, we see the transmitted beam out of our sample. So we have to be very careful while preparing the sample. So again we have a electron beam. So electron beam is coming and getting focused with the help of magnet lens over the sample holder. So this is a sample holder. We keep the sample over here, which is coated with which is uh, coated on a very thin TEM grid, which is made of sometimes carbon or copper, and this beam is getting transmitted from the sample and again diverged on this window. So this is a phosphor coated window, as I already gave you the example of a TV monitor. That kind of same screen is over here. We can see these electrons are coming on this screen, giving us the signal, magnified image basically. So this is the negative and this is the positive. So the electron moves from the negative to the positive. And then we have a vacuum inside. So we use a very efficient pump to maintain the vacuum inside this column. So this was invented by Ruska and others, and Ruska also received the Nobel Prize in the 1989 for the evolution invention of the TEM. So this is a typical anatomy of the TEM. We have a gun that is having a filament, and then the anode and condenser lenses, then objectives, intermediate lenses, and projector. So this is a normal. Uh, 1 second. Yeah. So this is a schematic of a normal slide projector. So what happens? We have a lamp over here, which is then the light will fall from the lamp to the condenser lens, and from the slide to the slide. Then from slide we have again having a magnification lens. With the help of that, we get a bigger or magnified image. So similar thing is happening here also, that we have a light source or the electron source and a condenser lens. Then on the specimen, this is for a normal optical microscope. In that case, we have a normal visible light. Then we have a condenser lens. Then the specimen. Then it goes to the objective, and with the help of an eye lens, we get a magnified image. So here we, in place of a light source, we have electron as a source, and that is focused with the help of different lenses. Then we have objective, and again multiple lenses and a projector. So with the help of that, we project the sample image on a fluorescent screen. So in the diffraction mode and image mode, we have we can do the imaging for the TEM. So what happens in the diffraction mode? We remove the aperture from here, and then we get the signal, which is in the form of a diffraction pattern. And in the image mode, we have a objective aperture, which uses a back focal plane, and with the help of that, we get a magnified image. That is the final signal. So that is the sole difference. The aperture we need to remove for the uh, diffraction, and then we have to add the SAD aperture. And in the image mode, we have to put the objective aperture and remove the SAD aperture. So these are the two modes in the TEM. Then we have the dark field and bright field imaging facility for the TEM. It will give the information about the internal ultrastructure, nanostructure dispersion, and defect identification also. And the high resolution HRTM, which is called, it gives us the information about the interface structure and defect structure. Then again, we also have the energy dispersive X-ray spectrometry, similar to the EDS as we have in the SCM. Here also we have the EDX. With the help of that, we can detect the elemental composition of the sample, and mapping and line scanning can be done. And then the diffraction mode again, that I already told you, which gives information about the crystal structure. And yields is also there. That this is one kind of spectroscopy which gives the information about the chemical composition and other bonding information. So that can also be checked with the help of TEM. So this is a typical TEM image of a uh, of this material, which is having silicon and copper oxide, and it clearly shows the interlayer between the uh, two materials. And at the high resolution, we can see the interlayer clearly, and and the atoms of the silicon and copper and their orientation can also be seen with the help of the high resolution TEM. So this is around 50 nanometers. We are able to see the alignment of the atoms clearly. So it is a multi-purpose machine. It will give us the information about the atomic structure. The diffraction is having a limited 1 ppm. So the resolution is very very high. It gives zero point 0.2 nanometer of resolution. And the microstructure and defects can be observed in the bright field as well as dark field imaging. It will give the information from the micron level to the nanometer levels, and the chemistry information also we can get. This is from the inelastic scattering. So X-rays, energy dispersive X-ray microanalysis, which is having a resolution of around 1 nanometers, and then electron energy loss spectroscopy EELS. These also can be uh, analyzed in the TEM. Then problems with the TEM. For polymers, it is little difficult because the sample preparation is one of the major problem because we need a very very ultra thin specimen from the bulk polymer, which is to get that kind of very thin specimen. That is a challenge. Then the polymers are very sensitive towards the electron beam radiation, and because of that, it can burn also. Since this contains organic substance, so that's why when we put electron beam, it sometimes damages the polymer. So we are not able to get the very good information, or sometimes we get uh, different information from the sample, or which is not useful basically because the material is already burned, so it will not give us the actual information about the sample. Then the contrast is also very low because here we have only very low atomic weight molecules, so those will not give us very good contrast. So that's why it is kind of difficult to obtain the TEM of polymers. So as I already told you, the beam damage, it causes the radiolysis. That is, it refers to ionization and breaking of the chemical bonds. So this happens in polymers, and then the knock-on damage. So after knocking on the sample, so the material will get damaged because the atoms are not displaced. So the crystal lattice and will be different. Like it will slightly um, get defective, and also the point defects will also be present in the metals. Then the beam damage can also be due to the photon generation. It causes by specimen heating, and it causes sample drift, and so because of the heating, the sample gets shifted. So and the drift occurs, and that's why we will not be able to get a better image with because of this. And the structure destruction can also happen because of the heating and melting. And charging is also one of the issues. In the SEM, charging is mostly dominant because for polymers, this charging happens because if a sample is not conductive, then it try to store the electrons. It will not uh, transfer the electrons. So in the for the conductive sample, the electrons get or charges gets transmitted or transferred to other part. But if a material is not conductive, then it try to store the electron. So electron will not go away from it, and that's why we will see very very bright sides at some point. So that kind of defect occurs because of this in the polymers because those are not conductive. Then cataloguminous. This is an optical and electromagnetic phenomena in which electrons impacting a luminescent material. So suppose we have a phosphor, so it causes the emission of photons. So if it is a fluorescent material, it will cause emission of photons. So because of that, the wavelength will vary in the visible spectrum, and that's why we'll not be able to get a very good image from the electrons. Then how to reduce this damage? So suppose if we are using a polymer, So in that case, we have to use very low EV electron volt of the beam. So in the SEM, we have option of reducing the power of the beam, and same with the TEM. So low dose technique, that is the shutter off method, we can use and with that we can image the material. And thin carbon coating can be done to restrict the static electricity and loss due to the volatilization and specimen movement. Then enhance sensitivity of imaging, more silver and light coated on the photographic plate, that is for the instrument. And then apply higher accelerating voltage and reduce inelastic scattering, that is one of the method. Then use scanning TEM, that is STEM. So this process, this uh, filtration technique is also can be used. Then cooling the specimen during imaging, and that means reducing the temperature, and then we will not be then that will not damage the material, or that will not cause the melting of the polymer. So then sample preparation and the elimination of noise. So when we talk about the sample preparation, I already told you that we need very thin cross section of the material, because the electron has to transmit from the sample. So it has to be very thin, and it should be around less than 100 nanometers. And it should be stable under electron bombardment in a high vacuum, and it must fit in the specimen holder. So the holder is having less than 3 mm in diameter. And sometimes if we have a nanoparticle, we have to coat it on the TEM grid. And then ideally specimen preparation should not alter the structure of the specimen. And also so research point of view, we have to see some of the literature search to get the idea of the uh, preparation of the sample, or methods which are appropriate for our sample. So techniques for final thinning. So if you want to thin our material, so for metals we use electro polishing technique. So in this, the specimen is made as anode and the electrolyte in an electrolyte cell, and then the current is passed so that the specimen is dissolved, and we'll get a very thin anode formation, film formation from the anode. Then ion milling. This is used for metals and ceramics. So a beam of very energetic ions is directed on a solid, and atoms can be knocked out from the specimen. Then we have ultramicrotomy. This is for polymers and biological samples. So a firmly mounted specimen is moved past a fixed knife and of glass and diamond. So with the help of that, we can cut the sample in a very thin uh, layer of film. So here are some of the images. So the sample preparation technique is shown here. So this is called as ultramicrotomy, and here you can see it is a method of cutting specimen into very thin slices. That means ultra thin sections, and this is mostly used for biological specimens, but sections of plastics and soft metals can also be prepared with the help of this. And it permits the internal fine structures of samples to be visualized and analyzed at nanometer scale resolution. So it produces ultra thin sections of material in a fast and cleaner manner. So as you can see here, the knife is attached. It can be of diamond or glass, and the sample is moving towards in this direction, and the sample is cutting. The knife is cutting the sample. So one of the technique is the wet technique at room temperature and dry for cryo temperature. So this is again a technique for the preparation of samples. Uh, so by making replicas for tea. So this is a single stage replica technique, then two stage replica technique, and this is extraction replicas. So this kind of techniques are also used, and then the hardening or fixation technique. So here we have a sample, and this is a tool, a knife, and water is flown, and it actually removes as a chip. The sample is removed as a form of a chip, very thin chip from the sample. So this is used. This is also a technique which is used for the preparation of sample. This is generally for metals. Then some of the errors. So these are some of the practical problems which are also there. So characteristics of the error and their origin. So periodic waves or different thicknesses are can be there because of the blunt or damaged knife edge and elastic sample, and there are various problems. And then we have single or several non-periodic waves. This is because sample is soft, extreme building vibrations, etc. And then changing thicknesses along an ultra thin section is because of the inhomogeneities present in the sample and in sectioning areas and wrong trimming. Then variation of thickness from section to section, so air movement and temperature fluctuations in the laboratory, or the section thickness attempted is very low, and strongly compressed sections because of the wrong knife angle or wrongly adjusted free angle. Then folds in ultra thin sections. This is because of the bad knife again, and very large sectioning area. Then impurities in section. Impure water used as floating liquid or insufficient cleaned aperture. Then folds in the section of a grid. This is because of the wrong floating and transfer on the section onto the grid, and sections are too large. So these are some of the problems which has to be avoided, like tried to be avoided to get a very good image. So alternate sample preparation technique is the spin coating or dip coating or solution coating casting on the grid itself, and in which is for the in situ microscopy, mechanical deformation, heating or cooling, electron irradiation, electrical or magnetic field, or different techniques also we can check. So for micromechanical, we can use the bulk polymers also for checking the mechanical property. So suppose we need to check the surface fracture. So in the tension test or impact test, or we can observe the changes, and then we can make the cut the sample from here and do the SEM or TEM from here. And then if we want to deform the sample, then suppose the cross section gets thinner, so from here we will take out the sample for TEM also and for SEM also, or AFM can also be done. Then we can also do the in situ deformation of very very thin samples. So we can put the sample in SEM and then uh, check, like apply a very small load and then observe the changes in SEM. Or again we can cut the sample from here and then see the SEM or TEM microscopy. So these are some of the grids which are used for TEM analysis. So these are different like variations are there. So bar grids, these are the bar grids, then mixed bar grids, then folding grids, and slot grids are also there. Hexagonal grids are there. Finder grids are there. Then support films, example carbon or polycarbon or silicon monoxide also. So these kind of grids are present. Then mesh is designated in divisions per inch. So this is 50 per 100 inch, 50 divisions per 100 inches, or 100 divisions per 100 inches. Likewise, these are designated. Then materials vary from copper and nickel to esoteric selections like titanium, platinum, gold, silver, etc. based on the demand or application. So the diameter of this grid is around 3 mm. So these are used for non-self-supporting specimens like powder specimens or quantum dots or nanoparticles. Then we also can stain the material to uh, bond with a certain molecule, and then we can observe the changes which are happening. So the functional group which are present in our material. So suppose we have this functional group, so if we have polymer that is polyethylene, so for that the staining agent can be chlorosulfonic acid, and so we have a normal aliphatic chain of carbons in this case, so it will react with it, and then we will get a proper signal out of it. So that is how the contrast can be enhanced with staining a particular material, staining the material with a particular agent. So also the etching can also be done. So polyethylene, for polymers like polyethylene, we use a etchant, that is this hot p-xylene toluene, or chloro this CCl4. So this is for the low melting species removal. So that's why the etching is done. Then for the laminar morphology and superstructure, if you want to reveal from a material, then this etching of permanganic acid with help of permanganic acid we can etch the polymer, and then the artifacts will get removed, and we'll get a better morphological image of the sample. So likewise, we have different etchants for the different polymers to get a better images. Then comparison again for the microscopic technique. So in the optical microscope, as I already told you, the size of the object is very high. So the point two or around 200 microns we can observe. Then in the SEM around uh, point 0.4 or 0.004 to 4000 microns we can vary the size or magnification. And in the TEM, the resolution is very high, around 0.0002, that is from 2 nanometers to 20 microns we are able to see. So here the depth of field in the optical microscope is very small, but in the SEM we are able to see the 3D structures also. So depth of field is good, uh, is large in the case of electron microscopy. Then objects, so in the optical microscope we can observe the surface or the bulk structure. In SEM also surface, only surface structure is preferred, and in the TM the bulk structure that has to be cut from the sample in a very thin film, that we can observe in the TM. Then the environment, in the optical microscope we use ambient environment, but in the electron microscopy we have to use very high vacuum to remove the noise. So that is one of the disadvantage in some samples, because most of the biological samples cannot be imaged in those cases. Then radiant damage. So in the optical microscopy there will be no damage, because we are using visible light as a source. But in the SEM there might be some damage because of the uh, high energy electron beam, and in the TEM we use very high electron energy beam, so it will be very uh, high, so damage will be severe in those cases. Then specimen environment, in this case, in the SEM and TEM, in the SEM and optical microscopy it is easy, but in the TEM we have to have very good sample preparation, so that is a difficult part. Then chemical analysis. So in the optical microscope we cannot do any chemical analysis or elemental analysis, but in the SEM and TEM we can do the analysis of the elements. Then detection of chain orientation. So in the optical microscopy we can observe the chain orientation. In the SEM we cannot, but in the TEM also we can observe the chain orientation, because we can see a particular atom. So in those cases we can see where the chain is initiating or getting terminated, those kind of things. So I've covered this, so I'll skip this one. So now let's come to the application of the TEM and SEM. So again, to check the fiber morphology. So in the optical microscope we were able to get a image of the fiber morphology, but in the SEM we will get even better resolution and better magnification as compared to the optical microscopy. So here it is showing an oligomer polymer or fiber surface, and the curved surface here of the fiber does not exhibit much detail other than the oligomer, and this is the oligomer particle which is shown by the arrow. And the micrograph in the B image, it is showing a similar region, and much detailed image can be seen other than the oligomer. So here we can see the depth of field basically. So that's why we can see a three-dimensional surface. So here we are able to see observe this part, and here we are able to observe this part, the other part. So this is basically because of the high depth of field which we use in the SEM. So fractures and crack analysis can also be done. So this is the image of a fractured fiber, and it reveals the flaws which are caused because of failure. And so this image one is the mirror, shows the flat mirror, and near the region M, and the adjacent to the locus of the fracture. So this is the locus or starting point of the fracture. And then this shows a very slow crack growth, and the fast crack growth region also, that is the region H. It has large ridges, and this is a magnified image of this particular uh, section, which reveals the flow causing failure. So the arrow shows the detailed picture of this. Then again, with the failure analysis, we can use it for different materials. So this is a nylon 66 fiber. These are the SEM tested at 0 to 55% of nominal breaking load, and the this shows the splitting of the fiber, and a very long tail is coming out of it because of the failure. And this is the very long shallow for of the other side of the fiber. So those kind of things can also be observed from the SEM. Then pre and post processing analysis. So this is a non-woven fabric. So figure A shows a side view of the fabric on the stub. You can see the thickness of the fabric over here. This is the SEM stub, and this is the thickness of the this is the sample. So this is the thickness of the sample, and then this is the top image, the face view or the surface of the mm, fabric. Then it will not give much information because it is a very like low magnification image. Then we can observe at a high magnification, we can see the different patterns. Like here we can see the patches kind of formation on this. So these are the local melting which are holding in the fiber altogether. So and again at higher magnification, the fibers are seen to arrange from round to the deformed shape. So this kind of post and pre-analysis, pre-processing and post-processing analysis can be done. So again, in the tire technology also, this has very good application. So tire cords are shown by several complementary microscopic methods. A cross section of an adhesive coated yarn is shown in the optical microscopic image. So this is the optical image. The surface of the RFL coated cord is shown by the SEM. This is the SEM image. Then comparative normal SEM and backscattered SEM. So you can see the difference in the resolution, right? So the normal SEM is giving higher resolution as compared to the backscattered one. The backscattered image shows the surface has higher atomic number particles. That means, as you can see from the arrowheads, those are giving better contrast and detailed surface structure that suggests that the adhesive is found in higher concentration in this interstitial regions between the fibers. So this is one of the analysis. Then the film and membranes can also be observed with the help of SEM. So this reveals a very porous structures in terms of the range of separation of applications. So The first one is showing the polyethylene membrane with elongated stretched porous regions. As you can see, these are the stretched regions. These are porous regions of different sizes, which are separated by fibrils in the while drawing, uh, while extrusion or drawing the sample, and unstretched lamina normal to the draw direction. So this is like showing the comparison. Then the surface structure is quite different from the three commercial. These are the commercial membranes, and this is the this they have, this is the elongated one, the drawn one. So this is giving the information about the how after drawing the material, how the morphology is changing. Then this is the cross section of a polysulfone composite membrane. So this A and B are the cross section, and C is the surface view of the material, and this is after etching, chemical etching of the surface. So it removes the active surface and it gives the bulk porous morphology. Then film and membrane. So the surface of this membrane, ETFE membrane, is imaged over here, and this is the surface. And when we see the at higher magnification, it is very clearly showing the this kind of morphology present in it. And the surface is appearing very wrinkled, as we see. The texture is wrinkled, and but imaging at lower magnification, after imaging at higher magnification, it did not reveal any this kind of contrast. So here like it will not give us very good contrast. And here I think this is because of the beam damage. This kind of thing is happening. Then film and membrane. So again, we can observe the films, two phases. Polymer system can be also imaged, and it reveals a porous, irregularly shaped, this kind of irregularly shaped pores, which is having very fine and dispersed particles, as you can see here. Then engineering resins and plastics. So we can also check the morphology of the resin and plastics which we have made using the SCM. So this first one shows the uh, complicated image of a network complex network morphology, and the B shows the etched surface, fracture surface, which shows the etchant has affected both the dispersed phase and the matrix. So various composites can also be analyzed with the help of SCM. Like we can see the different morphologies, the different processing after processing of the sample. So again, the image processing also has to be done. So normally we get a grayscale image. So the grayscale optimization and the shading and contrast, those kind of things we have to do. With this can be pre-process, post-process also after getting the data out of the SCM or TEM. So suppose sorry, these are some of the images. So after So these images are like when we have lesser resolution, this kind of image will be seen. But if we increase the uh, basically if we increase the pixel size, then we will get a better resolution with higher pixel size. And then we can change the contrast also. So as you can compare the contrast here, this can be done, and this again. The quantification can also be done. So the quantification of the different particles which are present at this particular image, so that can be done. Checked the diameter of these particles. That can be observed after pre-processing. So this can be done with the help of software, that is ImageJ software, or different like the ImageJ is very is like freely available software, and it is very easy to operate. So that is what generally people use, and we can use different softwares also for this. And Fourier transform. So the Fourier transform also can be done using this image software. So as we can see, if we want to see the spacing between these two, so we generate a Fourier transform image of this like this, and then we can check the spacing uh, after the from this grayscale profile. We can uh, after the processing we can observe the like in that case we will be able to observe very uh, accurate uh, image, like accurate gap. So from here we are not able to get, because the resolution is very very high, like the size is very small, so we will not be able to resolve it at that much position. So that's why we use this Fourier transform mechanism. Uh, we completely transfer the image in the Fourier transform, and in after that we are able to get the distance or the spacing between the two lines or the two planes. So similarly for different like different materials, we can do this Fourier transform analysis, and then we are able to observe the spacing between the two lines. So again, let's see, here you can see a ABS material. So these are having this kind of arrangement of the planes, and then we get a Fourier transform image. This is taken from the uh, TEM machine diffraction pattern. We can take, or also the Fourier transform it also can be taken. Then we observe the like different using like different FFT image. This is called as FFT image. This is the original image, then this is the FFT image. Then we inverse it to get the Fourier transform and filter it so to get the FFT image. This is the like post processing of these images. Then impact modified polypropylene. This can also be checked in the TEM. Then for polymers, this is for the specifically for polymers. So suppose this is a amorphous polymer. So it is a globular structure which is difficult to evaluate using scattering techniques because of the poor electron density. So hot deformation or surface etching or chemical attack, strain-induced contrast can be done for this kind of materials. Then we have the deformed SAN polymer. This is a craze-like deformation zone attached ahead of a crack tip in this copolymer. And so this figure A shows a overview, and this is at the largest magnification. You can see the crack tip and this zone, craze-like zone over here. Then different materials like polycarbonate can be observed. This is a deformed polycarbonate, and at different like magnifications you can observe the changes. Then we get that this kind of this is called as a diffraction pattern, which we get from the TEM, and this also gives us the information about the uh, like the arrangement of planes or the crystal morphology. This is the again the diffraction contrast you can observe. So you can see the planes very clearly. These are visible. Then for different blends of polymers, you can observe the patches for different materials like this blended. So that can be seen. Different polymer blends can be observed. So this is taken from the AFM tapping, and this is from the TEM metal graph. You can observe the changes. Then for different, so we can vary the concentration of the blend with the, and then we can see how the composition is changing in particular, and we can optimize the composition of the particular blend after checking the TEM images. Then rubber modified images, this can also be checked. Then block copolymers. So what kind of a structure these are making, those information we can get. So this is 28% PS in PS cylinders. So this kind of morphology is present over here in these cases. Then we have lamellar morphology. Then we have again cylinders. So this, like, the morphological change can be observed with different chemical composition or molecular composition. So again, the block polymer, which is having a star block copolymer, having a rich phase of butadiene, and which is appearing dark, and the PS particle are remaining unstained, so these are appearing bright. In different rubbers, like the natural rubber and butadiene rubber, if we are mixing it and thermally etched to get this kind of morphology. Then CB and silica in the SBR we can get over here. Then cross sections of different polymers, these are already told you. These are some of the slides which are repeating. Then the HRTM of graphene is very well studied from the TEM. With the help of TEM, we'll get a very good hexagonal pattern in the reflection pattern, which gives us the information about the structure of the graphene sheet. That sheet of that can also be observed. So that graphene sheet will be having very few sheets, like two or three sheets. This will give us a very good diffraction pattern like this. Then epoxy based nano composites can also be checked. So different. So if we are adding this nano clay in the epoxy, it will be very distinct in the TEM. We can observe the morphological changes after addition of the this clay, nano clay. So now coming to the question. So transmission electron microscope is eligible to analyze bulk morphology or defects in the structure or elements or all of the above. So can anyone answer this question? Yes, all of the above. Exactly. So we can observe the bulk morphology by cutting a very thin, ultra thin cross section of the polymer or material, and then defects in the structure. So since we can observe the atoms, then in the material, so those atomic defects can also be observed, and the elemental from the X-ray characteristic mapping that can be observed. So all of the above. So the second question is, the reason behind ultra vacuum used in electron microscopy is to get effective number of electrons, optimum speed of the electrons, uncontaminated surface, and source and sample, and all of the above. So what will be the answer for this one? Yeah, exactly. So with the help of the high vacuum, we will get a very good number of electrons imparting on the sample, and the speed also we can optimize, because in the vacuum the electrons are not getting distracted or uh, de-accelerated because of the air particles. So we will get a better speed of the electrons, and the sample is also uncontaminated because we are keeping it in a vacuum. So still the sample, it will not be having any contamination. So all of the above is the correct answer. Then which of the following software is generally used to process the microscopic images? So Origin or Mendeley or Image or all of the above. Can you answer this? Yes, Image. So this software is very widely used and it has lot of functions and is free also. So that's why it is a very good software for the processing of the images. And which of the following conditions will provide the best quality images of AFM in AFM? Slower scan rate, lower gap between the tip and sample, and faster scan rate, lower gap between the tip and sample, slower scan rate and higher gap, or faster scan rate and higher gap. What should be the answer? Yeah, exactly. So if the scan rate is slower, then we will get a better quality because the speed will be less. So we'll get more pixel points, and also the lower gap between the tip and sample. So the lower the gap, the more data, like the more deflection we can observe. Because if the gap is higher, then if it is deflecting, if the cantilever is deflecting, we will not be able to observe much changes. But if it is lower, then the tip is near to the sample, that means we are getting better results. So silica gel is used to control the humidity, temperature, or the atmospheric pressure of the chamber, or all of the above. What should be the answer for this? Okay, I'm afraid. I think the humidity is the correct answer. So silica gel are used for the are actually it soaks the moisture from the environment. So that's why it is basically used for the controlling the humidity in the chamber. So in the normal in bags and all, we get a, like if we purchase a new bag, we get a silica gel, this small pouch or small packet of silica gel. So that is to control the humidity inside the object. So that's why, because it absorbs the moisture in it. So that's why silica gels are used for controlling the humidity in the chamber. So option B is the correct. So with this, we can conclude today's session. So do we have any doubts? Depth of penetration. Uh, in what case? Like in which case you want to calculate? Okay. So depth of field or depth of penetration. So there is a formula which is like delta equals 1 by uh, under root 2 by something. I'm not sure about it, but we have to also know about the material property of the polymer. So if we know the properties of a material, like its conductivity, its permeability, then we can calculate. But otherwise, it is difficult to say. Yeah. Yes, roughness will, like if there are hills and valleys, then the depth of penetration will be high. Like if the valleys are there, then that means the we can able to image the valley deep inside the valley also. So that's why, like I can draw it. So suppose we have roughness like hills and valleys kind of this things. So the roughness, what did you say? How much is the roughness? So this height is 100 nanometers suppose. So the depth of field in this case will be 100 nanometers or more can also be there. So what are the options? Yeah, so clearly it should be less than 100 only because the roughness is 100, right? So the height between like the distance between the height and valley, the peak and valley is 100. So the depth of field will be around in this dimension only, like from 100 to maybe it is 50 to 100, or it can be from 100 to 150. I'm not sure, but I think it should be less than 100. Yeah, because the depth of field, so this is basically suppose we are putting the light from here, the beam, electron beam is coming from here, like so it will image this depth of the valley also, right? And then uh, if we move towards the this peak, then the depth will be this much. So I think that depth of field will be counted from here to here, so it will be around 100 should be something like that. Yeah. So normally in SEM, yeah, we get the surface information only. We are not getting from the bulk. If you want to check the bulk information, then we have to cut the cross section, and then we will get the image of the bulk. So this is mostly from the mm surface, but you are asking the depth of penetration. So I don't think like there might be a formula for this, but I'm not sure. I have to check the depth of penetration. But like right now I can think of this only, but I'm not sure. And also, yeah, lateral, what did you say, lateral displacement. Yeah. Okay. Mm. I think it should be the first case, because if the material is very very hard or very rigid, In that case, sometimes that starts this in the lateral displacement happens, but I'm not sure. Again, I have to check. Any other questions? Okay, if not, then we can conclude the session. Yeah, hello. Yeah, Upati, you are asking the question. Yes. Yeah, yeah, yeah. Five. Mhm. Actually, in that case, there was some problem with the options because the 10 to the power something was not matching with our solution. So maybe there was a problem in the options. So maybe you can ask. So when there will be a live session with the professor, that time you can ask him like what should be the correct answer for this. I am not sure about it. You can directly mail to NPTEL if this is the case. I can't help in that. Yeah, yeah, yeah. Okay. So if more number of people will say this, then they will. I think they will consider. So they will. Yeah, two questions were having problem, right? I remember. Yeah. So I think we have to you have to contact them. So after that only they can do that. So I can't help in that regard. Okay then. So there is no question. Then we can conclude the session. Thank you.